Welcome back, everyone, to the deep dive. You know how much we love to dig into the nitty-gritty of cutting-edge science, and today is no exception. We're tackling a question that I think has captivated humans for millennia. What makes us age?
It really is one of those fundamental mysteries, isn't it? And, aging is a process that touches every single living thing.
Exactly. So where do you even begin to unravel something that complex?
Well, a good place to start is by looking at the building blocks of life, our cells. If we can understand what drives aging at the cellular level, that can have some really far-reaching implications.
I like that. Start simple, right?
Exactly. And that's where today's deep dive comes in. We're focusing on a model organism that's, well, it's pretty unassuming to look at, but incredibly powerful for research. Baker's yeast.
Saccharomyces cerevisiae, our tiny friend from baking bread and brewing beer.
That's the one. But don't let its humble culinary role fool you. Yeast has been absolutely crucial in helping scientists understand the fundamental processes of aging, particularly in eukaryotic cells. Those are cells with a nucleus, like the ones we have.
And correct me if I'm wrong, but when we're talking about aging in yeast, there are two main ways scientists study it, right?
You got it. There's replicative lifespan, which is essentially about how many times a yeast cell can divide before it conks out. You can kind of think of it like how many copies a cell can make of itself.
Okay, that makes sense. And the other one?
That's chronological lifespan, or CLS. This one looks at something a bit different. It's about how long a population of yeast cells can survive when they're not dividing anymore. They're kind of in a suspended state, just hanging out.
Ah, so it's not about how many times they can reproduce, but how long they can persist in a non-dividing state.
Interesting. And that's what we're really zeroing in on today, right? Chronological lifespan.
Precisely, because we've got a fascinating new preprint to explore, and it dives deep into the genes that control this chronological aging in yeast.
Okay, lay it on me. What's this preprint all about? The title is a bit of a mouthful, but it gets right to the point. Exposing the core genetic drivers of chronological aging in yeast cells. It's by Cruz Bonilla and a whole team of researchers. Now, scientists have been studying the genes involved in yeast CLS for a while now, but this paper tackles a really interesting challenge that's kind of been hanging over the field.
Okay, I'm intrigued. What's the challenge?
Well, you see, when scientists do these big, large-scale genetic screens to look for genes that affect how long yeast cells can survive when they're not dividing, the results haven't always been consistent across different studies. There's been surprisingly little overlap.
Huh, that's unexpected. You'd think the same genes would pop up if they were truly important for aging.
You'd think so, right? But the thing is, the false positive rate in some of those earlier studies could have been pretty high, like 50% to over 90%. That means a lot of the genes initially identified as playing a role in longer or shorter lifespans. Well, they didn't actually hold up when other researchers tried to replicate the findings.
Wow, so only a small fraction of those supposed aging genes were actually consistently found across different experiments. That sounds like it would make it really tough to figure out what's truly going on.
It definitely complicates things. And the authors of this preprint point out that some of those inconsistencies might come down to things like what kind of food the yeast were grown on, the media differences in their genetic backgrounds, and even variations in the ways they were measuring lifespan.
Right, those experimental details can make a huge difference. So how did these researchers try to get a clearer picture of the core aging genes?
That's where things get really interesting. They took a really clever approach. They did a meta-analysis.
A meta-analysis. Okay, I'm familiar with the term, but could you give a quick refresher?
Sure. Basically, instead of just looking at one study in isolation, they gathered data from a bunch of different large-scale studies, 9 to be exact, that had already investigated how deleting specific genes affects yeast chronological lifespan.
9 studies. That must have been a mountain of data. Did they include everything they could find?
Well, they did leave out one smaller data set just to keep things more focused. But overall, they analyzed a whopping 45779 deletion strains.
Wow, that's massive. You definitely can't accuse them of not having enough data points. But how do you even begin to make sense of that much information?
They used a method called Prometheus. And what Prometheus does is it lets you not only see whether deleting a gene had any effect on CLS, but it also weighs how strong and consistent that effect was across all those different studies.
So it's not just about a yes or no, did it affect lifespan, but also about the magnitude and reliability of the effect. I like that.
Exactly. And this helps separate the real deal, the genes that consistently make a big difference in how long those non-dividing yeast cells live, from the ones that might have just been a fluke in one particular experiment.
So like weeding out the background noise to get to the clear signal? Makes sense. And what did this Prometheus analysis reveal? What were the top contenders, the genes that really seemed to drive longer lifespan?
Some of the biggest winners, so to speak, were genes like TOR1 and GLN3.
Those names ring a bell. Didn't we talk about TERA signaling before in the context of aging?
We did. It's a major pathway that's all about sensing nutrients and regulating growth. And dialing down TR activity has been linked to longer lifespans in a bunch of different organisms, not just yeast.
Yeah, I remember that. It often comes up when people are talking about the benefits of calorie restriction.
Exactly. So it's really interesting to see TR1, which is a key component of that pathway, come up as a top hit in this meta-analysis. Just reinforces how important the TR pathway is for regulating lifespan.
Okay, so TR1 is a big one. What about GLN3? What does that one do?
That one's involved in how yeast cells respond to the availability of nutrients and different kinds of stress. It's all about adapting to the environment and making sure the cell has the resources it needs.
Fascinating. So these are some of the genes that, when you get rid of them, the yeast actually live longer. What about the flip side? Were there any genes that stood out as being essential for longer life? Ones that, when you remove them, the yeast have a shorter lifespan?
Absolutely. RM15 was one of the standouts. And then there were a bunch of genes involved in autophagy.
Okay, autophagy. Remind me what that is again.
Think of it as the cell's built-in recycling and cleanup crew. It's the process that gets rid of all the damaged bits and pieces that build up inside the cell.
So it's like taking out the cellular trash.
Exactly.
Yeah.
And you might think that having a faulty garbage disposal system would make the cell live longer, but it actually seems to have the opposite effect. It seems like keeping the cell clean and tidy is crucial for long-term survival.
Makes sense. If the junk piles up, it's probably not good for the cell's health in the long run.
Precisely. Now, something else the researchers looked at was whether these genes that affect chronological lifespan, whether they do anything else in the cell besides influencing aging. It's this concept called pleiotropy, where one gene can have multiple different roles.
So it's not just about lifespan. These genes might be multitasking.
Exactly. So they looked at how well these different gene deletion mutants could grow, especially under stressful conditions or when they had to rely on respiration for energy. And they found that a lot of the mutants with shorter chronological lifespans also struggled to grow in those tougher environments.
So it seems like some of these genes are important not only for surviving a long time in that non-dividing state, but also for being resilient and thriving in general.
That's the idea.
Yeah.
But here's the thing. They also found that many of the genes that clearly affected chronological lifespan didn't seem to have any obvious effects on growth. So it's not a one-to-one relationship.
Right. So there are some genes that are specifically dedicated to keeping those non-dividing cells alive and kicking.
Exactly. And this actually ties in nicely with another question they investigated. We've talked about chronological lifespan, but remember, there's also replicative lifespan. That's the one about how many times a cell can divide.
Right, like how many cellular offspring it can produce. So did they find any connections between the genes involved in those two types of lifespan?
They did. And this is one of the really cool findings of this study. They found a pretty significant overlap.
So there are some genes that are important for preventing both types of aging.
Exactly. For instance, mutants with short replicative lifespans were more likely to also have short chronological lifespans, and vice versa.
That makes a lot of sense, doesn't it? If a gene is really crucial for keeping a cell healthy and functional overall, you'd expect it to be important whether the cell is busy dividing or just hanging out.
Absolutely. And they even pinpointed some specific cellular processes that seem to be shared between the two types of aging things, like autophagy, which we've already talked about, the organization of the vacuole, the breakdown of proteins, and even the machinery responsible for producing proteins.
So these are like the fundamental pillars of cellular health that support both types of lifespan. Okay, so we've been talking a lot about yeast, but I'm curious, do these findings have any implications for other creatures? Like, could they tell us anything about aging in more complex organisms, even humans?
That's the million-dollar question, isn't it? And the researchers actually addressed this head-on. What they did is they took all those yeast genes they identified as being important for CLS, and they checked to see if those genes have counterparts in other animals, like worms, flies, and even mice.
So like looking for those same genes, but in different species.
Exactly. And then they looked to see if those animal genes have also been implicated in aging in those organisms. It's a way to see if those aging mechanisms discovered in yeast are also at play in other branches of the evolutionary tree.
And what did they find? Are those same aging genes doing similar things in other creatures?
Well, here's the really exciting part. They found that the yeast genes, whose deletion led to a shorter chronological lifespan, Those genes were much more likely to have counterparts in animals that are also involved in aging.
So it's not just about yeast. It seems like those same genes are playing a role in the aging process across a whole range of species.
It really does suggest that there are some fundamental core aging processes that have been conserved throughout evolution. And that's a pretty profound insight, because it means that what we learn in yeast can actually provide some really valuable clues about aging in more complex organisms, including us.
That's incredible. So it's not just about extending the lifespan of yeast in a lab dish. This research could potentially have implications for understanding and maybe even manipulating the aging process in humans someday. Okay, so let's take a step back and think big picture. What are the key takeaways from this meta-analysis? What are the major cellular functions and pathways that they pinpointed as being really important for chronological aging?
So for increased longevity, one of the most consistent findings was reduced activity in the mTOR signaling pathway. We've already talked about that one quite a bit. But they also found connections to processes involved in DNA replication and the production of ribosomes. Those are the little factories inside cells that make proteins.
So basically, slowing down some of those core cellular processes might actually be beneficial for lifespan in the long run.
That seems to be the case, at least when it comes to chronological lifespan in yeast. On the flip side, they found that processes like respiration, mitochondrial function, autophagy, vacuolar function, the ability to respond to stress, and even gluconeogenesis, that's the process of making glucose from non-carbohydrate sources, those were all really important for longer lifespan.
It sounds like there's a lot of crosstalk and balancing going on inside the cell, doesn't it?
Absolutely. It's not just about turning one thing up or down. It's about maintaining a delicate balance between all these different processes to ensure the cell's long-term survival.
It's like a complex cellular symphony. Now, I know the researchers didn't just stop at identifying these genes and pathways. They also delved into the regulatory mechanisms, right? Trying to figure out what controls the activity of these chronological lifespan genes.
You got it. It's like trying to identify the conductor of that cellular symphony. They use a tool called tFrank to identify key transcription factors. Those are proteins that regulate the expression of other genes. And they found 46 of them that seem to be important in controlling the network of genes involved in chronological lifespan.
46, that's a lot of conductors for our cellular orchestra. Were there any familiar names in that list?
Yeah, some of them were already on scientists' radar, things like BAS1, TEC1, GEAS1, and some components of the HAP complex. But here's the really interesting part. Many of these transcription factors hadn't really been studied in depth in the context of chronological lifespan before.
Oh, so it's opening up a whole new world of research possibilities.
Exactly. And they also made a distinction between what they call global and local regulators.
Okay, break that down for me. What's the difference?
Basically, the global regulators are like the master conductors. They seem to influence a whole bunch of CLS-related genes across different pathways and functions. The local regulators are more like section leaders. They primarily control genes within specific aging-related pathways.
Got it. So there's a hierarchy of control, with some transcription factors having broader influence than others. Now, the preprint dives a little deeper into one specific transcription factor called TEC1. What made this one so interesting to them?
TEC1 seemed to occupy a kind of middle management position in that regulatory hierarchy. It's connected to a bunch of different aging-related functions, especially ones involving the mitochondria and autophagy. And to really understand what it was doing, they did something called a lifespan epistasis screen.
That sounds pretty intense. What exactly does a lifespan epistasis screen involve?
It's basically a way to see how two genes interact with each other in terms of their effects on lifespan. So they'd mess with TEC1 and then mess with another gene known to be involved in chronological lifespan, and then they'd see what the combined effect was on the yeast's lifespan.
So like a double whammy to see how those two genes work together.
Exactly. And one of the key findings from this screen was that TEC1 and mitochondrial function seem to promote longevity in parallel pathways. It's almost like having one working well can compensate for deficiencies in the other.
Oh, interesting. So if TEC1 isn't functioning properly, healthy mitochondria can help pick up the slack and vice versa.
That's the implication. And they also found a connection between TEC1 and the cell's ability to sense nutrients, which ties back to that whole TOR signaling pathway we were talking about earlier.
Wow, it really does seem like everything is interconnected, doesn't it?
It definitely does, and I think that's one of the biggest takeaways from this study. It highlights just how complex and interconnected the aging process is. But at the same time, it also gives us a much clearer picture of some of the key genetic players and pathways involved. And the fact that some of these findings seem to hold true in other organisms is incredibly exciting. It suggests that we're really starting to unlock some of the fundamental secrets of aging.
I completely agree. It's amazing to think that studying a tiny, single-celled organism like yeast can provide such profound insights into a process as universal and complex as aging. And it makes me wonder what other secrets are hidden within these tiny cellular universes just waiting to be discovered.
That's a great question to ponder. Who knows what other fascinating insights are just waiting to be uncovered as we continue to explore the intricate world of cellular biology.
And that's what makes science so captivating, isn't it? It's a constant journey of discovery, and with each new finding, we get a little bit closer to understanding the grand tapestry of life.
I couldn't have said it better myself.
Well, on that inspiring note, it's time to wrap up today's deep dive. But before we go, a huge thank you to all of you for joining us on this journey into the fascinating world of chronological aging in yeast.
Yes, thank you all for listening. We hope you enjoyed this exploration of cutting-edge science as much as we did.
And please don't forget to like the video and subscribe if you want more content like this. We're always diving deep into the latest scientific discoveries, and we can't wait to share them with you.